Today we're going to be talking about cell division part 2. So at first we talked about mitosis. And with mitosis we were talking about making identical copies in the offspring or the daughter cells. And in meiosis or meiosis, you could think of it as meiosis made me, it's how we make offspring cells or sperm and egg cells that are not identical to the parent cell. And we call these sex cells. They're going to have half the number of chromosomes. So when the product of these are made, they're going to be half the number. So if we're talking about humans, a diploid cell will be 46. All the sperm and egg cells will be 23. And the name we have for that is haploid. So these are going to be produced in the ovaries in females and the testes in males. So what is the actual purpose of these processes? Well, mitosis, this is going to be general growth and development. This will happen with our skin, our bone cells, brain cells. Meiosis is going to be sexual reproduction is the purpose. So with mitosis, it'll go through one round of division, producing two identical daughter cells. Meiosis is going to go through two rounds of division, producing 4 cells, and all the offspring are going to be genetically unique with half the number of chromosomes. And the name we have for these cells that have half the number of chromosomes, our sex cells, we call those germ cells. And your regular cells that have a full number of chromosomes, we call those somatic. So comparing the two processes together, if we look at mitosis, well, the DNA is doubled in S phase, and then in M phase, when it splits, it's going to go back to having the same number of chromosomes. Whereas when we talk about meiosis, the DNA is going to double, but it's going to go through 2 rounds of division so that the offspring have half the number of chromosomes as the parent cell. Here's another image of that, is referencing the chromosome number. So, one way to remember meiosis is we talked about mitosis, and mitosis was PMAT. That's how we remember that process. Well, meiosis is PMAT times 2 with a couple of key differences. They're going to help lead to genetic diversity. That's one of the big benefits to meiosis. So just looking at the general framework here we'll skip forward and go to each step, but let's back up a little bit. So the same thing happens at the beginning with prophase, where the DNA has to condense from chromatin into chromosomes. But the key difference in prophase 1 of meiosis 1, because again there's 2 rounds of a division, one of the key differences that leads to this genetic diversity is crossing over. And this occurs in prophase 1 of meiosis 1. And in mitosis, the DNA condenses into chromosomes, and they don't interact with each other. But in meiosis 1 of prophase 1, the chromosomes actually overlap gene coding regions, and they swap genes in the same areas. And depending on how often these genes swap, we call that the crossover frequency. And that crossing over creates completely genetically unique chromosomes by swapping genes from one side to the other. And that's going to lead to a lot of genetic diversity. But generally, everything else is going to be the same: prophase, metaphase, anaphase, telophase. And we'll look at some other differences. But they call that pairing, they call it a synapsis or a synapse. So during this event, homologs exchange sections of DNA at specific points, and they call that chiasmata. So these chromosomes, the way this leads to genetic diversity is that the lining up and the swapping of genes is completely random, which means every sperm and egg cell is going to be completely genetically unique, which creates a lot of genetic diversity. And then when we pair that with the fertilization that will take place between this genetically unique sperm cell and a genetically unique egg cell made by a female, at fertilization, well, now we have 2 completely unique sets of DNA coming together, and now you have an even more unique offspring. So genetic diversity is a huge plus to these processes. So then we'll go into each step. So prophase 1, biggest thing here, biggest difference is going to be that crossing over occurring. And that's what differentiates it from prophase of mitosis. So all the other same things occur, right? Spindles form, centrioles. And then in metaphase 1, the homologous chromosomes that are paired will now line up at the equator for separation. Anaphase, they get homologous pairs, get pulled apart. And again, homologous pair would be like 2 of the X's. So they'll get separated to each side. Then we get into telophase, which is where we start to form 2 new nuclei. And you start to see 2 new cells forming. And then in cytokinesis, you'll have that cleavage furrow compress all the way through and separate it into 2 cells. So here we see meiosis 2, and we'll look at those steps. So meiosis 2, again, you have prophase 2 again. So this new spindle is going to form, and we've got these chromosomes in the condensed form. Metaphase 2, the chromosomes line up at the equator. Anaphase 2, they're going to be pulled apart. And in telophase cytokinesis, we're going to end up with 2 new cells, but we do this for both cells that we got from meiosis 1, so we're going to end up with actually 4 cells. So we'll go through some of this. Now, this division will contain 2 cells in each stage, so that adds up to a total of 4. And again, it looks just like mitosis, it just happens twice. So we go to prophase 2. We got 2 cells in prophase, going through this on both sides. Meiosis 2, metaphase 2, line up in the metaphase plate. Anaphase 2, they start to get pulled apart. Telophase 2, we start to form 4 cells, because it happens twice, which is going to make 4 haploid cells with half the number of chromosomes. And then there, it's just walking you through that full process until the end, where you end up with 4 genetically unique cells with half the number of chromosomes, so that, let's say if this is a sperm cell and it goes and fertilizes an egg cell, well, then you get 23 plus 23, and you get 46 again at fertilization. So we have different types of meiosis. We have in males spermatogenesis, and in females oogenesis. And one is to form sperm, the other is to form egg. In spermatogenesis, of course, this is going to occur in males. You have this one primary spermatocyte. It divides during meiosis 1 to form 2 secondary spermatocytes, and then that will eventually develop into 4 spermatids. And the 4 spermatids mature into 4 spermatozoa, or mature sperm. So with males, it's make bukus of sperm cells, as much as could possibly be made. And in females, it's about making a quality egg instead of quantity, because the sperm are going to come to the egg. The female reproductive system is creating an environment that fosters the most fit sperm to find the egg in that ampulla of the fallopian tubes coming out of the ovaries. That's actually where fertilization occurs, and the sperm has to swim against the flow in that region. So the most fit sperm will get there, and the key for oogenesis is to make the most fit egg for the sperm to fertilize. So from all of this, it makes these secondary oocytes and then these polar bodies. And in the end, you end up with one supreme premium egg for the sperm to actually interact with and fertilize. So here's a comparison. The oogonium, as it develops, you end up with one really good egg cell. Spermatogenesis, we end up with 4 sperm cells. So quantity, and the egg strategy is quality. So for fertilization development, for example, we have gamete formation. So gamete is a sex cell. So gamete formation will be the production of those sex cells. Fertilization is when the sperm and the egg meet. So some basic terms you should be familiar with. And we call that first fusion, that fertilization, we call that the zygote, or the first cell of a new organism. And then we talk about cleavage. The zygote is going to undergo several cleavages to where new cells are going to be produced as it goes through differentiation. And then gastrulation, these cells are going to specialize into tissues and tissue layers. So there's different types of fertilization. Essentially, you have fertilization that takes place outside of the organism and fertilization that takes place inside of the organism. So for external, the sperm and the egg meet out in the environment. So we see this with fish. External fertilization with amplexus, the male and female embrace amplexus as the sperm and egg are released. So we see that with frogs, maybe toads. So the sperm and egg still meet in the environment, but the male and female are actually embracing each other. But it's still external fertilization. Internal fertilization, the sperm is placed inside the female, and this can happen in a variety of ways. We see this with like birds, for example. But internal fertilization with penis would only occur, of course, in more formed organisms that have that structure, such as mammals and reptiles. And the sperm is going to be placed inside the female using the male genitalia. So again, fertilization, sperm meets the egg, and we go from 23 and 23 to the zygote with 46. And from that point forward, it's going to start growing and reproducing and cleaving, and it will start to develop really quickly. So the cleavage, this results in a hollow ball of cells called a blastula. And you'll see as it goes through that the size of the developing embryo doesn't change that much in that first few hours of development and first few days. It's not really getting that much bigger. It's just cleaving a lot. So here's that process. It goes from a zygote to a two-cell stage, 4-cell, 8-cell, right? Each cell is dividing, so it's doubling and doubling until eventually we get to a morula, which is that cluster of 16 to 32 cells. Then it goes to a blastula, where it gets more hollow, and then an early gastrula. And as we start to get into these steps, we start to develop these, well, if it's triploblastic, these 3 germ layers, depending on what kind of organism it is. And then the gastrula at the end, we start getting those tissues beginning to form from differentiation. So here's a slide with some images of these forms highlighted. So number 1, that's an unfertilized egg. Then number 2 here, we have a fertilized egg, so a zygote. 3, you'll notice it's 2 cells right next to each other, so it's that first 2-cell stage. And then we've got a 4-cell stage. And then 5, we've got about 16 to 32, so it's probably a morula. And then number 6, you can see that it's hollow on the inside, so that's going to be a blastula. And then number 7, you'll notice this little crevice that's coming in, this depression. That's how you know that it's a gastrula. And that little opening is called a blastopore, and that can develop into either the mouth or the anus, depending on what type of organism that it is. So that gastrulation will result in 3 layers, up to 3 layers. We have the ectoderm, which is ecto, think like outside. The mesoderm, meso is the middle. And then the endoderm, think endo, inside. It's that inner layer. And these are going to develop into, the ectoderm will end up being more of the skin of the organism. Mesoderm, a little bit deeper, maybe muscle tissue. And then endoderm might become more of the internal lining of the organs and the digestive tract. So as these cells develop, they're going to differentiate. So certain genes are going to be turned on, certain genes are going to be turned off as they become the types of tissues that they need to be to construct the organism in question. And we call these cells, before they differentiate, we call them stem cells. So they can become any type of cell. And I believe that is it for today's lecture.